I think now it'll be great if we can just have a session where we have free dialogue, and I have a few questions that I'd like to pose to the panel. And maybe I'll start with you, Aviv, given the fact that, we've now heard these multiple talks about the plasticity and regulation of the immune response. How can we hope to integrate the power that you showed us with the single cell RNA sequencing and other approaches, of course, the computational approaches, but try to also capture the biology of this dynamic process so that even when we're looking at specific genes that may say, okay, this gene is the hallmark gene for a repressive program, but maybe that gene is actually not being expressed because the protein expression may be very different as the cell is going through its plasticity in its different states. How do we try to integrate the different technology advances to get better at capturing that dynamic process of the immune system?
So that's a fantastic question. I'll start with the fact that one of the strengths that the computational technologies give us is that we don't actually have to put all our hopes in one or another gene when we try to understand what's going on, but we can work at this level of multiple genes together, a program, or as I showed, even multicellular programs across multiple cell types. The result of that is that we get actually a signal that is more robust to the, not just to the differences in the biological levels of regulation, RNA versus proteins, but also just in the capabilities of the experimental assays to give us the most precise information. So by looking at the level of multiple genes and big profiles, we actually get more robust answers. This, of course, then switches when we have to start thinking about, say, therapeutic development, because there we actually do need to choose specific things that we want to go after. And I think it plays out in two different ways. When we go after a molecular therapeutic, you need to find the mechanism. It's not enough to just say there are cells of this kind. You need to figure out what is actually going on in these cells. And that's where we need to triangulate from three different perspectives. One is the patient samples that really figured prominently in Zemin's and Miriam's and my talk, because we have to start with the human biology and the human as the model. Then the ability to do experiments in a dish with patient-derived models like cells co-cultures, organoids for some tumors. That's very advancing very rapidly. And finally, of course, use the animal models as well. And by moving between the three of them, we can add interventions and perturbations. It can be drugs that we treat with, but it can also be genetic manipulation. And that allows us to understand the molecular causes. So that's I think the second perspective. And then the third one that comes back is when we try to do therapeutic development with those targeted therapeutics that we develop, we come back to the same approaches that we started with, because now we can look at the human patients in a much higher and more sophisticated resolution that we were afforded before. Before, you did have to choose a single biomarker. And if that choice had any limitations, technical limitations, the signal was not as great, it kind of worked, but it wasn't the perfect representative for all aspects. That was it. Now we can work with much richer things as our biomarkers, and that helps us understand better what is happening in the patients when we treat them and stratify them in more sophisticated ways and so on. So I do think it plays out in different ways at different points along the process of understanding what is going on, but it puts us in a different place than we were before. And the technologies keep getting better. Single cells, that's like so 2011. Now most of us are using spatial analysis approaches so we can see how they relate to each other. Just doing RNA, again, with clinical samples, you had to collect them 5 years ago in order to have answers now. So that's what people see today. But if you ask us about the experiments we do today, it's RNA and protein and the organization of the chromatin, multiple things all being measured together. So the technologies advance extremely rapidly right now. And that means that we would be able to know a lot more quite soon.
So I think this brings us to the point that you said about humans, right? You have to start with the model system that's going to be the most meaningful, and getting those samples becomes so important, and getting them from all of the different places that we talked about, right? They're not just a primary tumor, but the metastatic lesion. Then is the metastatic lesion in bone? Is it in brain? Is it in liver? Because these are all very different niches. So Miriam, maybe you can talk to us a little bit about how do we make that happen? Because you've put together this great team, that we saw in your slide, but how do we make that the forefront of everything we're doing? There are over 2,000, maybe even 3,000 clinical trials right now that are ongoing with immune checkpoint therapy agents, have we really brought this to the forefront of the pharmaceutical companies and the academic organizations that reading out the clinical endpoints without putting together these teams that can give us access to these samples, we're doing ourselves a disservice that way. How do we make this better?
Yeah, so it's a very good point, and now Aviv, could you potentially even comment on that, because she's has crossed, to the other part of the aisle.
And, I'm going to get back to her.
Exactly, but I will even say, something we say now is that it's almost unethical not to do ancillary study or biological studies on these clinical trials, because as we know, and Aviv will comment on that much better than me, is that most phase 3 trials fail, and unfortunately there's a lot of failure in these phase 3 trials. In fact, the way we called our group was a group that will help. The way we define our group that is engaged into this immune profiling of clinical trials, and now we are doing it, we are using the same type of group to go and profile patients during treatment, is that this will help us de-risk the phase 3 trials. It's really, in fact, we have a little commentary that's going to come up in Nature Medicine this week, where we describe that, not only in cancer, Pam, right, across medicine, because in cancer, I think the industry is much more advanced. Like they agree, they're so they realize there's so many molecules, they need a little bit of help from biologists to really make things make sense of all these different type of immune molecule. But in other disease, so I'm trying to do the same in inflammatory disease, and there is much more resistance, right? So you will do all these very expensive clinical trials. You are going to bring a new molecule to a disease. You have the potential to use this brilliant technology that Aviv has discussed, which will cost probably a fraction of the cost of the clinical trial. And yet, there is resistance from industry. So I think industry has to step up, and they are in cancer, less so in other disease. The other things is that our group, there are other resistances, sometimes the surgeons or the pathologists. The pathologists are extremely, protective of their tissue. And when they protect to store the tissue, not so much to really do a lot of research on the tissue. So I understand there is legal procedure you have to bring and you have to store the tissue, but there is so much you can do with this tissue. So we have to change the mindset of our pathologists. They have to realize that they need to share. They need to share with scientists that, of course, are going to make a good use of this tissue. And then the third thing is we need to think of what are the relevant tissue pipelines, because sometimes the tissue is not informative, right? When it has been bombarded with many different things, maybe this is not a tissue to study. I am asked to do a profiling on very heavy radiation, and I'm saying, well, this time point are just going to collect dead cells. Don't bother the patients with the biopsy. So we need to brainstorm relevance of the tissue. Pathologists need to be on board, surgeons need to be on board, and industry need to step up because it's extremely expensive. And NIH is not, as we all know, NIH is not really also stepping up enough. They think that profiling has been done enough and we don't need to profile anymore. These are the type of critiques we have, in our NIH grant asking for profiling tissue. So it's a complex ecosystem. We need to change the culture. This technology is extraordinary. We need to bring it to the clinic and not leave it on the bench of, sophisticated, scientists. Need to bring it to the clinic.
Yeah, and I couldn't agree more. And Aviv, as you know, we've started these kinds of studies back in 2004 when I ran that first bladder trial with 10 patients and got so much pushback. How do you think industry can be brought to the forefront to say, hey, before you do an 800 patient trial, let's do a 10 or 20 patient small study that gives us information? How do we get the Genentechs of the world to, really fall behind us and support this concept?
We definitely do that. We are a research and early development organization to begin with. And so our studies are phase 1 and phase 2 studies. They are smaller, and our goal is really to understand, right? To know whether a drug is safe, of course, and then efficacious, but to understand it in high detail. And so this work for us is actually at the heart of what we do. But I will say more generally for Genentech and Roche is that we've been in the forefront of this kind of work. You know, going back to the days where there wasn't really single-cell RNA sequencing, it was still mostly about genomic sequencing. We include consent for genomic research analysis in every trial. You often can't go back, even if you have the perfect trials with the perfect samples collected. If you have not consented the patients appropriately, you know, tough luck now, there's no going back. And I actually have to be grateful to the people who came before me for having had the foresight to think about some of these things 5 years ago, 10 years ago, when it was less common to do trials like this, so that now we can come back to those samples and ask questions years later when the trial completes in the right way. And I strongly agree with Miriam's point about the importance of doing this across many different diseases. This is important for each disease on its own, but it's also, we have learned so well that disease mechanisms repeat themselves in very unexpected ways.
Absolutely.
CD20, very important to target in cancer, very important to target in MS. Two very different diseases, a big B cell component in both. That's a big lesson for a company like Genentech, of course, but it's a big lesson, I think, for us as a community, that you never know with biology exactly where the mechanisms would be. So everything that allows you to look across diseases strengthens you.
Especially in the immune
other
system, right?
things.
Especially with the immune component that is going to be present across the cell
And
that are populated.
stromal cells and your epithelial cells, it's just true. And I mean, you're preaching to the choir with me on this and with my colleagues. We have learned, though, some important lessons. Samples are limiting. The better the technology is at getting you more in one assay, the better off everyone is, because there's so many different things that you can do. Which one would be the most impactful? In the end, the sample will be limiting. It's a biopsy. It's very small. There's only so much you can do with it. So that's where technological advances are important. And the clinical world is not the experimental lab in a research institute. Absolutely. So all sorts of things you can arrange in a very convenient way. They fall away when it's a global clinical trial with dozens of sites, and you work by proxy through those sites until the samples are collected. And so you have to think very hard as a technologist, as a computational person, as a clinical expert, as a biological expert, to really make an operation like that successful. And that's where I think the partnership is so important, both in the early phases, as Pam highlighted, doing the small study right sets you up on the path to success. And in the late stage, when you have the very big trials, as Miriam referred to, doing those in a way that comes back and restarts the cycle of discovery so that we can find more, because, you know, there's still enormous amounts of unmet needs. And so that's for us a big commitment in Genentech. We have new departments for that, cross initiatives for that, lots of collaborations for that. And I hope others will do the same.
Absolutely. We need more people to follow Genentech. But let me just highlight the CD20 example that you give. So Zemin, maybe you can join this conversation, because I saw your data set about CXCL13, and I think that's very intriguing because, as you know, it's very much a chemokine that's going to attract B cells into that microenvironment to have that T-B cell collaboration, tertiary lymphoid structures. So do you think that maybe we need to start looking now at these cell neighborhoods in a more careful manner to dissect how each of these cell subsets and what's driving their trafficking into the microenvironment, whether it's hormonal, BMI, neuronal, all of these different signals that we have to start looking at?
Yeah, absolutely. I mean, so if you don't simply study the single cell without the neighborhood context, without the spatial information, maybe we are missing some of the key information. But now if you combine a single cell with the spatial data, it should actually become clear. I didn't have the chance to present some of our work here related to that, but TLS, the tertiary lymph structure is actually very important for tumor in terms of regarding how they respond to different external therapeutic perturbation. And so those are, I think, going forward, if you look at CXCL13 is one of the manufacturers, but you can probably use different cytokines to somehow perturb or maybe enhance the TLS so that that location can be the place for generating more kind of pro-inflammation signals. So I think our affiliate company, Abiosciences, is actually taking some strategy toward this. I'm sure there are other places that are doing this as well. I think, but I really want to, going back to the previous question, Aviv and Miriam just mentioned about the having different pharmaceutical company involved in understanding human tissues. And we, when I worked at Genentech for many years, but when I started my lab in Peking University, I found out actually it's very relatively easy from my end to get clinical samples, and we were able to form the kind of really tight relationship with companies like Amgen, Boehringer Ingelheim, and Bayer to really collectively study those clinical samples. We're able to get a large number of really high quality data, which are very beneficial for finding tumor-specific subtypes and therapeutic targets. And so those are kind of things that can happen in a pre-competitive space. And I imagine multiple companies like Genentech and Gilead, should probably participate in things like that, so that we can generate a huge amount of data that can be used. We have already shown that, in today's several talks, that such single cell and spatial data can be extremely powerful in identifying tumor-relevant cell types and targets. And I think that this can happen definitely in a pre-competitive space, and some kind of correlation may be needed going forward.
Yeah, we're going to have larger data sets, so then we'll have to figure out how to get everyone to share their data sets, because that's the beauty of the strength in the computational algorithms, right? The larger the data sets, and then just bringing it all together from all the different groups, sharing it in a way that we can all use it. I think these are all pieces that we still have to put together. I'm going to switch.
The problem, Pam, with industry, though, Aviv, I have to say, there is a need for a push to, putting the data out there. I have to say, even when you guys publish in journals, we are asked to put all our data out there, and industry is giving a pass. And I think that somehow this is not so useful.
I would just speak to Genentech. I can't speak for the industry as a whole, but Genentech publishes its data. We write our papers and we publish our data. And in that, we actually are often an outlier compared to a lot of others. But
we need that to be broad across.
I am a big believer in that as well, but I cannot speak to everyone.
Let me take a few questions from the audience now, if that's okay with everyone. Aviv, this is, I think, for you. Can we extend these new aspects of immunotherapy and systematic approaches towards liquid cancers, like lymphomas and blood cancers too?
So I did not speak to that, but it's not just can we, absolutely, and people have and are doing it actively. I'll highlight one additional technological features that can be applied in many liquid cancers and is harder to do in solid tumors. Well, first of all, they're often easier to monitor sequentially because of the systemic nature, and so easier to obtain samples and follow multiple times. But second, there's a lot of questions related to clonality in the cancer cells themselves in hematologic tumors. And their single cell analysis, which is a genetic analysis, has all sorts of extra tricks that you can apply. For example, we have shown that you could use the mitochondrial genome and transcriptome in order to identify clonal mutations that have nothing to do with the cancer itself, but helps you identify the clonal relationships inside the tumor and how they evolve under treatment, which is tremendously beneficial for understanding both the original tumor and mechanisms of resistance. So that is an example of work that has been done, and lots of these efforts were led by Vijay Sankaran from Children's in Boston. But there is a lot more, and these approaches fit very well, very, very well in that world.
Okay, well, I think maybe I'll try one last question, Miriam. If we give a short answer, we'll see. What is the duration of time needed to fully understand the mechanism of tumor formation and resistance for a given cancer? And I think it's an important question, because sometimes we're looking at a single time point and it really is a duration of time that forms that. But maybe a brief answer before we finish up.
Well, that's a complicated question that we are all struggling with. I mean, we need to understand this dynamic of tumor, but this it goes back to our modeling question that we were asking is how do you best model? I think the way to approach it is what I think Zemin was also referring to, is really trying to understand, going across tumor, across different tissue type, and really trying to build, this commonality of cues, which he identified so well. And that will help us understand, some pattern that seems to be always present, as he showed, or whether, patterns that are specific to tissue type. And this is how we are going to build this big puzzle. These are complicated questions that many of us are addressing experimentally in animal models, much more difficult to address in patients.
So Janine, I think I'm going to turn this back over to you at this point.
Yes.
I think it's been a really robust discussion. Thank you.
Absolutely. Thank you. I just want to say thanks so much to you, Pam and Aviv, Miriam, Zemin, for a really fantastic session today. The talks and the discussion were really rich, and I'm sure everybody really loved it. Thanks as always to our audience for joining us from around the world. Please take a moment, if you have one, to complete the very short survey that I've linked in the chat box. We always look forward to your feedback. And don't forget to join our next Life Science Across the Globe event, which is taking place on April 6th at 9 a.m. Eastern Time, on the topic of new chemistry for biological systems. And that is being hosted by NCBS in Bangalore. So we hope you'll all come back to join us.